I want to talk a little bit about safety risk. Christian, I'd like to start with you on this one. Achieving production at adequate scale is a safety profile dependent endeavor. Why is improving the safety profile of mRNA therapeutics so challenging? What's challenging about it?
I'm not sure if I would agree to the statement you gave. So if you say it's either a good scale or good quality, that might be true for one and the same process. So you have always to compensate between yield and quality. But if you compare different processes, this might be a different story. So I think that the goal would be to have a scalable process where you do not have to trade off quality. And I think that's the goal where we have to go for. And the way how Ethris is seeing that. I think in general, players in the field have done a great job until now, and there is no reason to expect that the safety profile of mRNAs in general would be problematic. I think there's clear evidence and literature outside that shows that a modified mRNA has clear advantages over a non-modified mRNA. But apart from that, speaking about modified mRNAs, the safety profile for the marketed vaccines has been demonstrated and proven, and doses have been applied to numerous people, and the safety profile was very, very well, I would state.
Yeah. Okay, Patrick, anything to add there?
Yeah, in principle, we also have to see how much data in humans we have, and the current data is mainly on vaccines. We have, I think, billion of doses applied, and what we see is, I think, a very good safety profile that's comparable to other vaccines. If you go down, how the mode of action of these things is that we are mimicking a viral infection in the muscle and also in the immune system. And some of the side effects or reactogenicity that's seen is the onset of the immune response. So usually if you have an infection, your immune system is ramping up, so you will have fatigue. The body usually also sometimes reacts with fever. Connected to this, you get headache. So in principle, if you want to trigger an antiviral immune response without any side effects, I think your immune system is not reacting. Yes? So you need some reaction to really kick on the process of producing this highly specified, specialized immune cells. For therapeutics, I think we have less data so far. There were some first trials on intravenous administration, but also here, I think the current scenario is that you can use messenger RNA also for these things. I think in the end, some of the side effects or safety profile will be driven by the purity of your materials. We touched this already on the production purification stuff that we discussed. I think we will also see in the future more and more data out of humans that might give us better understanding on what is driving which side reactions.
Yeah. I hadn't planned on asking about like raw material safety and safety in the context of development and manufacturing, like lab worker safety. But we had a couple of questions come in regarding that, so I want to put those out there while we're talking about safety. You know, we're talking about patient safety, but I want to talk about the safety of your colleagues because, like I said, we had some questions come in from the audience on that. Nicole, for instance, was interested in your thoughts on the containment of the equipment for worker protection during mRNA scale-up. And Jeffrey was sort of related. Jeffrey was interested in hearing about cleaning carryover limits and methodology around preventing cross-contamination. So I guess, you know, sort of two different questions there, but regarding safety in the lab development and manufacturing processes.
I would take this question. principally, usually it's like this, that you have to protect the messenger RNA from your workers, not the other way around. Because the messenger RNA is quite unstable, especially if it's not complexed already in the lipid nanoparticle. So you have actually to implement protection to condemn the messenger RNA away from your people, just to protect it that it's not get degraded. So usually you are really working under sterile conditions, and also the people have protection stuff on, but it's really to protect mostly the messenger RNA, not the other way around. Forgot what the carryover was the other question. Sorry.
Yeah, the second question was, around cleaning carryover limits and methodology around preventing cross-contamination. I think that plays to the point you just made, Patrick, that it's more about protecting the product than it is the people.
So that is also adding to the versatility. You can principally, if you wash, for example, with a very basic aqueous solutions, you will destroy the messenger RNA. It will be hydrolyzed. And usually there is not really a carryover. So usually in the GMP, you could principally use your equipment after washing for the next lot of maybe even a different product. At least this is the things that were in the past approved by the regulatory authorities and during the manufacturing license, because it's really clear that there is the possibility of carryover is quite low.
Okay. Christian, anything to add there?
I think the only thing to add is that similar to other bioprocesses, mRNA can be produced in single-use systems, which, if used, further reduce the risk of carryover, because you just produce your batch and use new material for the next one.